All right. Hey guys, welcome back to my channel. Today I'm going to be going over neuromuscular junction. And so I'm just going to start off by drawing the neuron in the muscle cell. So here's the neuron. Muscle cell. And then when I'm all done drawing, I'll go over more of the anatomy of the cell and what each part of it is. Alrighty, so we have here, this is the neuron, and on the neuron are some channels that we'll go over later on. Inside the neuron are vesicles filled with the neurotransmitter acetylcholine, and this is the axon terminal. And this space right here is called the synaptic cleft. And this is the muscle cell right here. And inside the muscle cell, this is the sarcoplasmic reticulum. This is the sarcoplasm. And these are the T-tubules, which are just like invaginations on the muscle cell. And here we find some more gates that we'll go over more as we go into neuromuscular junction. And so this picture here demonstrates voluntary muscle movement. And so this is a skeletal muscle cell. And so our bodies have different types of muscles. And this is the muscle that we move voluntarily. Because there's some muscles like within our organs that we that our body just does on their own involuntarily, that our brain doesn't tell our muscles to move, they just do it. And so this is voluntary muscle movement. And so it starts off with the brain sending an action potential down the neuron. So that action potential is the opening of these voltage-gated sodium channels. And so a voltage-gated channel is closed, but it will open when there's a change in voltage. And so sodium will start to flood in to the neuron, causing the neuron to become more positive. And that positive voltage will then open this channel here, which is called a voltage-gated calcium channel. So that channel will open, and now calcium will start to flood into the neuron. And when calcium floods into the neuron, it will bind to something called a snare protein, which is attached to the vesicle filled with acetylcholine. And so this dot that I'm drawing here is going to be calcium. So that is binding to the snare protein, which causes the vesicle to diffuse into the membrane of the neuron, causing exocytosis of the acetylcholine. So the acetylcholine is getting excreted from the neuron. The reason why that's important is because this is a chemical synapse. So the neuron communicates its signal through the use of chemicals, neurotransmitters, in this case acetylcholine. So acetylcholine starts to fill up the synaptic cleft. And I forgot to draw for a second. This is acetylcholine esterase. And we'll talk about that in a minute. But these right here are ligand-gated cationic channels. And so these channels open with a voltage, but these channels open with a ligand or a trigger. And so in this case, the ligand is the acetylcholine. So that will open these channels, and they're permeable to cations. So cations will start to flood in. And cations are basically just any atom or ion with a positive charge. And so an example of a cation is sodium. And sodium has more protons than electrons. That's why it has a positive charge. Other cations are potassium and calcium. And so they will flood into the muscle cell, causing the muscle cell to be more positive. And acetylcholine can also be eaten up by these Pac-Man looking things called acetylcholine esterase. And acetylcholine is made up of two things: acetate, which is excreted and then used by the body, and then choline is then recycled back into the neuron through the choline transporter and recycled again. So that's what those Pac-Man things do. But anyways, back to the muscle cell. So these cations are flowing through, making it more positive, which then triggers this voltage-gated sodium channel, causing another action potential to go down the T-tubule. And then that opens this voltage gate, which is called DHP. And DHP is mechanically gated to ryanodine. So if this is DHP and this is ryanodine, whenever DHP opens, ryanodine opens as well. And ryanodine is the gate to the sarcoplasmic reticulum. And the sarcoplasmic reticulum is filled with calcium. And so when ryanodine opens, calcium will start to flood into the sarcoplasm. And within the sarcoplasm is a molecule called actin. So I'm just going to start drawing actin. One circle of actin is called G-actin, but then they can make up an actin strand, which is called F-actin. And so on the actin strand is an active binding site. And that active binding site can bind to a myosin head, which causes muscle contraction. But we'll say in this scenario that calcium is not present in the sarcoplasm, or there isn't a lot of calcium. And so when there isn't calcium, this F-actin strand is blocked by a protein called tropomyosin. So that tropomyosin will block those active binding sites. And then there's another protein connected to it called troponin. And so when those active binding sites are blocked, the myosin head cannot grab on to those sites, and so there's no muscle contractions and no movement. And here's the scenario with calcium. So there's those active binding sites. Calcium will come in to the sarcoplasm. There's calcium, that little dot, and it will bind to troponin, which then lifts up tropomyosin off of those active binding sites. So now they're open, and this myosin head now can bind to them, causing the muscle to move this way, which is also called a power stroke. And so that causes muscle contractions. So when there's no calcium, the tropomyosin is blocking the active binding sites, and there's no muscle contractions. When there is calcium, it lifts up it binds to the troponin, which lifts up the tropomyosin, and now the active binding sites are open, and myosin can now bind to it, causing power strokes. So yes, calcium, yes, muscle contractions. But there's different ways on which you can break the neuromuscular junction, and I'll just talk about a few right now. So let's say that someone took a drug that inhibited these cationic channels, which are also called the nicotinic type 1 channels. And so if those channels were blocked, those cations would never flow into the muscle cell, not causing it to be positive. So these positive charges would not be here, which would never then activate this voltage-gated sodium pump, which would never activate an action potential down to DHP, which would never open ryanodine, and calcium would never flood out. And then calcium would not be able to bind to troponin, lifting up tropomyosin. So basically, if these channels were blocked, the active binding sites on the actin strand would be blocked as well, and there'd be no muscle contractions. And another way that you could break it is through something called a circuit pump. So this is the circuit pump, and the role of the circuit pump is to pump low concentrations of calcium from the sarcoplasm into the sarcoplasmic reticulum. And so let's say if this someone were, like if this sarcopump was overactive. If it was overactive, it'd be pumping a lot of calcium from the sarcoplasm into the sarcoplastic reticulum, and there wouldn't be a lot of calcium in the sarcoplasm, which would then result in less muscle contractions because there's no calcium to bind to the troponin, lifting up the tropomyosin. So that's what happened if the sarcopump was overactive. But if someone were to block the sarcopump, then there would be no way for the calcium to be pumped into the sarcoplastic reticulum. So there'd be a ton of calcium in here, which would then cause way more muscle contractions because there'd be a ton of calcium being able to bind to troponin, lifting up tropomyosin. And so there'd be way more muscle contractions. And yeah, so that's basically neuromuscular junction. Let me know if I missed anything or if there's something I should work on. So.